Welcome to The Explainer. Today we're going to talk about a piece of tech that completely changed, well, pretty much everything. Biology, medicine, even how we solve crimes. We're talking about the DNA photocopier.
So just to give you an idea of the scale here, imagine you have to find one single typo, not in a book, but in a library packed with millions of books. That's literally the kind of challenge scientists were up against for decades when they were trying to study just one gene inside our massive genetic code.
And then, boom, 1985, a totally revolutionary solution shows up. It was a technique so powerful it could find that one word, photocopy it 1 billion times, and basically just hand you this giant stack of paper. This thing is called Polymerase Chain reaction, but you probably know it as PCR.
All right, so let's get into it. Before PCR came along, trying to zero in on one specific gene was, well, it was a nightmare. The amount of genetic information is just so massive that finding one tiny piece of DNA was painfully slow, and a lot of the time just plain impossible.
So what PCR did was solve this huge problem of scale. It's basically a biological photocopier. You give it a microscopic, targeted piece of DNA, and it just amplifies it, right? It makes so many copies that it becomes super easy to see and to study. It takes that one little genetic needle and turns it into its own giant glowing haystack.
Okay, so how does this thing actually work? How does this amazing biological photocopier do its job? It's actually surprisingly elegant. It really just comes down to a simple recipe and a repeating cycle of heating and cooling.
So you start by mixing 4 key things in a teeny tiny tube. First, you've got your original DNA. That's the template, the document you want to copy. Then you add something called primers. Think of these like bookmarks or search terms that tell the machine where to start and stop copying. Next up is Taq polymerase. That's the real workhorse, the enzyme that actually builds the new DNA. And finally, you need the ink for the copier, the nucleotide building blocks, the A, T, C, and Gs. The whole shebang happens inside a machine called a thermal cycler, and it just repeats a simple 3-step cycle over and over. Step 1, denaturation. The machine heats up to about 94 degrees Celsius, and this melts the DNA, splitting the double helix into 2 single strands. Step 2, annealing. It cools down a bit, which lets those primers we talked about find and stick to their target spots. And finally, step 3, extension. The temperature goes back up to 72 degrees, and that's the green light for the Taq polymerase to start building brand new strands of DNA. And that's it.
Now, this is where the real magic happens. Every single time that 3-step cycle finishes, the amount of DNA you're targeting doubles. So you go from 1 to 2, 2 to 4, 4 to 8. It's not just adding, it's multiplying. It's exponential growth. And get this, after just 30 or 40 of these cycles, which only takes a couple of hours, that one single invisible molecule of DNA becomes over 1 billion copies. 1 billion! It takes something you couldn't possibly see and turns it into this massive, unmistakable signal.
Okay, so the basic PCR photocopier was obviously a massive breakthrough. But science never just stands still, right? So pretty soon, researchers started giving it some incredible upgrades to make it even smarter and more powerful.
The first big upgrade is something called real-time PCR, or qPCR for short. The best way to think about it is like adding a copy counter to the machine. You see, standard PCR just tells you at the very end, yep, the DNA was there, or nope, it wasn't. But qPCR, it actually measures the copying as it's happening, cycle by cycle. So it doesn't just tell you if the DNA is there, it tells you exactly how much you started with. A huge difference.
But what about that typo problem we started with? Finding a single incorrect letter in our genetic code? Well, these tiny variations are called single nucleotide polymorphisms, or SNPs for short. They're super common, but spotting them can be really, really tricky.
And that is where another cool technique comes into play, high-resolution melt analysis, or HRM. Here's how it works. After all the copies are made, the machine slowly heats the DNA back up and very, very carefully watches how it melts or unzips. It turns out that even a single letter difference changes the exact temperature at which the DNA strands separate. So HRM is like this super sensitive proofreader that can spot that one tiny typo.
And you can really see how powerful this is when you look at how it deals with different kinds of SNPs. They're grouped into 4 classes based on how hard they are to tell apart. Now, class 4 SNPs, they're the toughest of the tough. The change is so subtle that they're really hard to spot. But a good HRM analysis, it can reliably pick out even these class 4 typos. That's just incredible precision.
So we've made the machine smarter.
what's next? Well, the next big leap for PCR wasn't just about making it more powerful. It was about making it smaller. It was about taking this amazing tech out of the big fancy lab and putting it right where it's needed, out in the field.
Yet researchers have actually managed to shrink that whole complex desktop machine down into a device you can hold in your hand. This is what we call point-of-care testing. It means you can do this advanced genetic analysis in a small clinic, at an airport, or even out in the middle of a farm.
And these little devices are an absolute game changer. For one, they're ridiculously fast. Some can run through 40 cycles in less than 10 minutes. Wow! They're portable, obviously. They use cheap, disposable chips, so you don't have to worry about contamination. And they can even test for a whole bunch of different things all at the same time. So when you step back and look at the big picture, this ability to find, copy, and analyze a tiny piece of DNA wasn't just some small improvement. It fundamentally rewrote the rules for modern biology, and way, way beyond.
I mean, its applications are literally everywhere. In medicine, we use it to diagnose infectious diseases, to look for genetic markers for cancer. In forensics, it's how you identify a suspect from a single hair follicle. Agriculture, it's helping us develop crops that can fight off pests. Environmental science, we're using it to track things like drug-resistant bacteria in our water. It's just everywhere.
So yeah, it is absolutely no exaggeration to say that PCR is one of the most important scientific inventions of the last 100 years. You'd be hard-pressed to find a life science lab anywhere on Earth that doesn't rely on it.
And that's the thing. As this tech keeps getting smaller, faster, and more powerful, the possibilities for what we can learn from just a single drop of blood or water or whatever, they're practically infinite. Which leaves us with a really exciting question to think about. When you can hold a genetic lab in the palm of your hand, what are we going to look for next? What new discoveries are right around the corner?